Hello everyone, welcome to today's live broadcast, In Vivo PET SPECT Imaging of Human Brain Neuroreceptor Systems, 3 Decades of Progress. I am Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's event. We have a few important announcements before we begin. This presentation has been approved for a Category 1 Continuing Medical Education credit by Loma Linda University. If you are interested in obtaining CMEs after attending this presentation, please click on the Get Your Free CME CE Credits button in the bottom left corner of the event screen. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the green Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Dr. Dean Wong. Dr. Wong is a professor in the Johns Hopkins Medicine Department of Radiology and Radiological Science. He also serves as Radiology Vice Chair for Research, Administration, and Training, and Director of the Section of High-Resolution Brain PET Imaging within the Division of Nuclear Medicine. Dr. Wong received his undergraduate degree in physics from the University of Western Ontario and completed his MD at the University of Toronto in 1977. In 1980, he came to Johns Hopkins as a Research and Clinical Fellow in the Radiology Division of Nuclear Medicine. He received a doctorate in Radiation Health Sciences from Hopkins in 1990. Over the course of his career, Dr. Wong has been a leader in the design, development, and use of PET scanning for the study of brain chemistry. He has been a primary investigator on numerous federal and private grants, has authored or co-authored more than 200 articles and 50 books, chapters, and essays, and has been an active consultant to pharmaceutical companies. In 2013, the Society of Nuclear Medicine and Molecular Imaging bestowed upon him the Paul C. Aebersold Award for Outstanding Achievement in Basic Nuclear Medicine. I will now turn it over to Dr. Wong for his presentation.
Very much for that nice introduction. Today I'm going to speak about in vivo PET and SPECT imaging of the human brain, which goes over approximately 3 decades of research that was initiated all over the world. And I was just fortunate at my early stage in career to be present during the beginning of what really was a wave of research that I'd like to share with you today. These are my disclosures, and they'll be online. What I wanted to indicate was that among the neuroscience techniques, there are a number of techniques which have much higher spatial resolution, such as pfMRI and obviously confocal microscopy, which are indicated on this axis, but the temporal resolution as well is higher, but the specificity for studying the living human brain and also a preclinical in vivo brain is not really matched by most techniques because of the neurochemical specificity of the studies that have been initiated with PET since the early 1980s worldwide. And I'm fortunate to have been a small part of that at Johns Hopkins, which I'll give you a little bit of background on. What's important is that we are now moving towards the millisecond range, and I'll very briefly talk about an initiative that we were fortunate enough to get funded as part of the White House NIH Brain Initiative at the end of this presentation. This slide indicates the various techniques, some of which you may be familiar with in terms of function. I know that many of you are familiar with fMRI and MRI. PET and SPECT is a form of functional imaging which allows us to directly measure metabolism with a higher sensitivity and typically a higher resolution than magnetic resonance spectroscopy. And initially, blood flow PET was really the gold standard by which methods such as ASL for MR are now taking place. In the 1960s and 70s, oxygen and glucose metabolism was the next role. And then from approximately 1983 onwards, studies in North America and then all over the world began with receptors and then subsequently pre, post, and intersynaptic measurements. And so as part of PET and SPECT, we study neurotransmitters, enzymes, precursors, and more recently, we're able to indirectly measure the intersynaptic changes of neurotransmitters themselves. Now, this is a picture to remind us that in the 1970s and early 1980s, as PET was in its infancy in its use in human beings, glucose metabolism was and still is one of the important factors, certainly in neuroscience, and has actually found an even greater role in areas of function, including oncology and cardiac studies. And of course, since this is a neuroscience lecture, I won't talk about that, but I just wanted to indicate that what I'm going to talk about today is not about glucose metabolism, but is about the next generation, which has been going on for the last 3 decades. That's neurotransmitter imaging. In order to do many of these studies, we use radiotracers such as carbon-11 and F-18, which have a 20-minute and 120-minute half-life. And these are, as you know, the elements of life because many of the important molecules biologically, especially in the central nervous system, include C12 and F18, or a small modification of it, as I'll show you in a minute, can be very useful for imaging. So because C11 and F18 are unstable radioactive substances, we have to make them in what many of you would refer to in the old days as an atom smasher. It's a cyclotron which makes radioactive compounds like this, and we got ours in 1979. Suffice to say that the F18 or the C11 typically are radiolabeled to the compound of interest. In a minute, I'll show you most of these structures are 400 molecular weight, and they're basically antipsychotics and antidepressants or derivatives of these, which have an unstable C11 or F18. These decay, as I said, between 20 minutes and 110 minutes. And when they decay, they give off a positive electron, which only travels about 1 to 2 millimeters in the body. And of course, in the brain, which is largely water, there are many stable negative electrons which make up our body. Through the conservation of energy, momentum, and mass, the positive and negative electron match up and they annihilate each other. And in order to conserve momentum, they give off 2 gamma rays, 511 kiloelectron volts in energy, almost exactly 180 degrees together at the same time. So it's our ability to measure these 2 sequences almost simultaneously that gives us the high sensitivity and specificity of PET for neuroreceptors. Here's an example of just a single case where a positron, say from a labeled antipsychotic, reaches an electron which is, say, in brain water. As soon as they match, they give off the 511 keV photons, which then are measured by 2 detectors at 180 degrees to one another. If the electronics detects these within a very short window, and now there are those scanners that can allow measurements for time of flight, so it can actually detect the slight time difference and then figure out where this came from. This can then be used in the following way. After you get many millions of these coincidences at various times in the brain, we can then see that each pairs of detectors can electronically determine where these measurements are. Now, how does this differ from CT scanning? Well, with CT scanning, as you know, we give an X-ray through the brain, and then they measure detectors in a ring. In this case, the radioactivity is giving out 511 keV photons, which initiated from the unstable positron, and it's the ability to measure these many pairs that then can then be back projected to the brain and figure out where the brain chemicals are in 3 dimensions. We are fortunate to have one of the 18 highest resolution commercially available PET scanners in the world at Johns Hopkins Hospital, and this is the HRRT, which currently is being overseen by, is being maintained by the Siemens company. This has many millions of coincidences at any time. It has the ability to provide as much as 2 millimeter resolution images. So as you can see here, this is actually a green monkey that is only 4 kilograms, and you can clearly see the putamen and caudate in these sagittal slices. And in the human brain, this happens to be a glucose measurement. You can clearly see the cortex and the internal capsule and other structures here on this FDG patient, which almost looks like an MRI scan because we have approximately 2 millimeter resolution. We actually, in this scanner, have 207 axial slices. The typical PET scans prior to this generation of scanner might only have 15 to 30, and we have 207, so we can cover the entire brain. And this just happens to be an example from a phosphodiesterase 4 ligand that measures with C11 roloprem all these different slices. So one can cover the entire brain and part of the cervical spine. The acquisition, reconstruction, and backup of these studies are complicated, and they involve not only acquiring all these various interactions, but the reconstructions can be as much as 30 gigabytes of data at one time. But we've managed to accelerate this using special computer clusters so that the reconstructions can take place within 4 hours, 4 to 6 hours. The original first HRT took 1 week to reconstruct a 30-minute dynamic study. So you can see we've really progressed as computation improves. Now, this is the key to what we're doing today, and I ask you to please look at this. There are 2 papers that are important from the 1970s that I think you should be aware of. One is by Philip Seaman from the Pharmacology and Psychiatry Department at the University of Toronto, and the other one is by Saul Snyder from our own Neuroscience department here at Hopkins. And they show similar things. This particular one shows on this axis the in vitro binding affinity measured by the IC50. And for those of you who are not so familiar with binding affinity, what that means is it's the amount of a drug that's needed to block 50% of the binding of a trace of a tritiated tracer. So in this case, it was tritiated haloperidol, I believe, against all of these different unlabeled antipsychotics. And in the 1970s, these were the whole bunch of them. So for example, the original one, promazine and chlorpromazine, is up here, and it had the lowest binding affinity. And on this axis is the range and average clinical dose for controlling patients with schizophrenia in milligrams per day. What I find fascinating about this slide is that this is still useful today, even in 2nd and 3rd generation antipsychotics, which have different antipsychotics on them. and even in antidepressants and other disorders. And so that the neuroscience of neuropsychiatric drugs are that there seems to be a formal relationship in this log-log plot between binding affinity in the test tube and actual controlling of some of the symptoms of schizophrenia. Now, of course, these are only some of the symptoms. We still don't have the ideal drug for schizophrenia yet, which I'll talk about later while we're working on this area. But nevertheless, for many of the positive symptoms of schizophrenia, such as active hallucinations and delusions, even back in the 1970s, this relationship occurred. The compound that we first started with, and not only we, but there were at least 2 or 3 other groups in the U.S., focused on spiropyridol, which was a very high affinity and, as you can see, very potent drug for controlling schizophrenia. But like so many things in science, there were complications, and I'll come back to that in a second. That spiperon allowed us to label D2 receptors, and now this is just a partial list of the many areas that we can look at, from dopamine, serotonin, opiates, nicotinic, alpha-4, beta-2, alpha-7, histamine, norepinephrine, and what I haven't put on this list is things like cannabinoids and glutamate, which I'll talk about towards the end of this lecture. This is a picture, courtesy of my colleague Hiroto Kubera at Hopkins, that shows a whole list of binding potential maps, for example, in dopamine, D2, in terms of dopamine, opiates, all the various types. This is OMAR and other compounds, serotonin transporters. So many of these compounds, all done in human beings, as over the last 30 years. So how did we get to where we're going now? Well, we were fortunate. There were many groups trying to fluorinate spiperon, which I showed you earlier, was the most highest binding, and at that time seemed to be one of the most potent antipsychotics. But we had a nice collaboration between Bob Daniels and Bengt Lindström from Uppsala University in Sweden. And Bengt and Bob had the idea that instead of labeling the fluorine, which is difficult, they tried to label this entire molecule by adding a methyl group. Spiperon itself does not have this methyl group. And so that's why it's called N-methylspiperon in the 3 position. And since we can make C11 methyl iodide with high amounts of radioactivity so that most of the atoms are labeled with carbon-11, between Bob and Bengt, they were able to, in 1982 and 1983, radio-label this and create this molecule. And then through a lot of preclinical testing, we confirmed that this is labeled D2 receptors the same way that that spiperon itself would label it. And in the past, spiperon in tritiated version alone without the methyl group was actually in the late 1970s and early 1980s the prototype tracer for in vitro studies. Now it's replaced by many compounds like raclopride and others. But in those days, this was the specific one. Now later on, other groups labeled the fluorine 18 and fluoromethylspiperon and others, and the rest is history. So we were fortunate at Hopkins that I was just a student then, perhaps at the level of many of you who are listening in now, and my former mentor, Henry Wagner, who passed away about 3 years ago, he volunteered to do the first study, and in May 1983, this was the radio-labeled spiperon showing the basal ganglia for D2 and D3-like receptors, and the cortical regions, which are primarily 5-HT2A and alpha-adrenergic. We now have much more specific compounds, but it worked, and we were able to move forward with this study. And so this is, if you want to read about it, it's from Wagner et al., Wagner, Burns, Daniels, and Wong from 1983. And I was fortunate to be part of this as both a student and a trainee. I had a little more hair at that time. And then exactly 1 year later, one of my colleagues, Jim Frost and Saul Snyder, the most recent director of the Neuroscience department at Johns Hopkins, in the Solomon Snyder Department of Neuroscience, and now Rick Hugener is the chair. But he came to the PET Center because it was his idea to radiolabel a derivative of fentanyl. And so carfentanil was made exactly 1 year later, and the first imaging of the opiate receptor occurred. And of course, we were able to block both this compound with naloxone and this compound with haloperidol. And I'll show some examples of that later. So here is an example of the dopaminergic system that shows all the things that we can label. And here we see that not only did it start with labeling postsynaptic receptors, such as the D2-like receptors with spiperone, but around the same time, in 1983, the group at McMaster University in Hamilton, Ontario, developed radiolabeled fluorodopa, and this allowed a measurement of dopa decarboxylase. Now, fast forward 30 years, we can routinely label the BMAT transporter of the vesicles. In fact, some of this is going to become eventually commercially available, we believe, in the U.S. In the 1990s, we developed with Michael Kuhar at Emory not only spiperone, but the radial labeling of the dopamine transporter, where, as you know, cocaine and other and methylphenidate and amphetamine can act in part, and it's the reuptake site for endogenous dopamine. And so that was labeled with a cocaine conjugate. And then, more recently, as I'll show you in a minute, even though we cannot measure dopamine directly, we can measure the degree to which dopamine displaces radiotracers binding either to the transporters or to the postsynaptic receptors, which gives a measure of intersynaptic dopamine. Here's a picture that shows what actually can be done. This is a 2-day study that was done in Tourette syndrome as well as restless leg syndrome, where we were able to measure high specific activity raclopride. That means that most of the radiotracers molecules are labeled with carbon-11. And then we added carrier or unlabeled raclopride, and you get 2 points on a scatter to measure receptor density. We measured on the same day, in the absence and presence of an intravenous amphetamine challenge of 0.3 milligrams per kilogram given 5 minutes before the second injection, the dopamine release that I referred to. And then on the second day, we were able to measure the dopamine transporter using this superscript 11C Win 35,428, which we developed with Mike Kuhar. So in 1 day, in 2 days, we were able to do dopamine receptor density, dopamine release, and dopamine transporters. In other words, we could get all of these things here in just 2 days. And that's a typical example of the power of this tool. Now, my colleague and friend Tony Grace, who was kind enough to invite me to be part of this wonderful webinar, has worked heavily on the role of tonic and phasic dopamine in his many research areas and hypotheses about neuropsychiatric disorders, including schizophrenia and substance abuse. Suffice to say, I'm sure you've heard about this from Tony already, but the tonic dopamine, or the homeostasis dopamine, is more extracellular and more a stable level. And so, for example, Dr. Grace believes that there's in schizophrenia there may be a reduction of dopamine due to hypoglutamatergic changes, which downstream changes tonic dopamine, and then that change, which might be more of a trait than state situation, results in this state issue, where when you have a changed dopamine level photonically, you lower the brake, or you lower the amount of inhibition of phasic dopamine. And phasic dopamine is what we believe correlates with burst firing of the VTA. And in particular, Dr. Grace believes that this phasic dopamine, which we might attribute to positive symptoms of schizophrenia, or some of the positive acute effects of reward with dopamine, may in fact be measurable with PET, as I'll show you in a minute. So this brings us together, the tonic and phasic story. And again, phasic dopamine, we think, is related to the measurement of intersynaptic dopamine that I mentioned a couple minutes ago, whereas the tonic dopamine, it's a little harder for us to measure with PET, but we think that controls how much dopamine is released, either in behavioral states or in the presence of pharmacologic challenge. So I want you to remember that when I talk about phasic dopamine in the PET studies, you'll have a molecular picture of what's going on. And in the end, this may be, in fact, at least in schizophrenia, affected by the glutamate system. Now, one area that I've had the opportunity to work in is Tourette syndrome, and you can read about this in our 2008 Neuropsychopharmacology paper. And this shows that when we do an amphetamine challenge, we're actually able to demonstrate when we give amphetamine, when we give about 3.3 milligrams per kilogram of amphetamine, we're able to demonstrate that we have release of dopamine that occurs here compared to the controls, which is the more of the red bars. And in patients with comorbid Tourette's, which happens at least 20 to 30% of the time in people with Tourette's, there's even a greater release. So we think that there is a combination of these which affect the lability of phasic dopamine. Furthermore, more recently, we've been studying OCD subtypes, and we find that people with Jerry Nestat of our center, who's an OCD expert, and we find that people with OCD aggressive and symptomatology, as compared to those people who are more concerned about germophobia, etc., there's a big difference between the amount of amphetamine-induced dopamine release in the presence of whichever OCD subtype they have. Now, what about behavioral stimulation? In behavioral stimulation, there has been a literature, even going back to the late 1990s, that dopamine release can be changed potentially by cues, and I'll show you evidence of this in a minute. And where different dopaminergic parts of the basal ganglia are active during early exposure to reward when reward is unpredicted, which of course many of you are familiar with, the Wolfram-Schultz hypothesis. And finally, direct evidence was shown by the Gatsby group in London that when video games were shown to young boys doing and escalating video games, they had more and more release of dopamine. So here's a SPM, or voxel by voxel map, showing the brain areas that lit up when young boys played a video game as they got higher and higher in that video game process. Now, this is a study that we did in collaboration with Edith London and the NIDA intramural program in Baltimore, and we published in 2008. And this is kind of exciting because we showed Craving, audiovisual Craving tapes to diagnose subjects with cocaine dependence. And we know that about 50% of people that are showed craving cues, that is paraphernalia and pictures of active drug use, such as in Baltimore anyway, it's making up smoking, snorting, or intravenous injection of cocaine. And there was a videotape that Dr. London and I used that originally came from Steve Grants, who published a study in FDG a few years earlier, also with NIDA. But in this case, we did the raclopride measurement, which is known to be affected by intersynaptic dopamine. And in this case, you can see we have the amount of release of dopamine, that is the displacement of dopamine in the presence of these craving cues in those people that did crave, and it's about 50% in Baltimore. But so about 50% are down here, but the 50% that did crave, they all gave visual analog rating scales of their craving in the PET scanner. And as you can see, there was a direct relationship between craving and dopamine release, or intersynaptic dopamine, which we think relates to phasic dopamine release. And a similar finding was found by Nora Volkow, the director of NIDA, with her colleagues at Brookhaven in the same year. Now, I want to say something about PET and its role in drug development. As many of you know, unfortunately, there has been a crisis for the development of psychiatric drugs, especially and to some extent neurological drugs, because less and less are being developed worldwide, and especially in the US, because the risks are very high and the costs are very high. And even now, of course, we have virtually no drug that is successful long-term for Alzheimer's, and many of the drugs for the other major psychiatric disorders, the number of new drugs being developed are smaller and smaller each year. And so this is a slide from Steve Paul, who's now at Cornell and was at Lilly many years ago. And he suggests that this is the traditional route and the cost of developing a drug, which is about $1 billion. But one way of reducing the cost is between the preclinical level and early phase 1 to try to kill off those drugs that are not likely to make it. Well, PET fits right into this space. We actually help determine dose levels and drug levels and determine which drugs should go on to the next step. Here's an original example of this done at our center, where a subject received N-methylscripirone with the binding to the basal ganglia, and in the presence of about 8 milligrams of haloperidol given 4 hours before the PET scan, you can see there's a big degree of occupancy or blockade. This is just like people filling up a stadium at the Super Bowl or filling up a hotel during a convention. And it turns out that Lars Farda and his team at the Karolinska, using raclopride, where raclopride was invented, discovered this almost axiomatic diagram, at least for first and second generation antipsychotics in schizophrenia. And that is that as you get to 100% occupancy, and as I showed you a minute ago, this is close to about 80% occupancy, 70% occupancy. As you get close to 100% occupancy, and you look at this against the oral dose of antipsychotics, because of course antipsychotics are given orally or by depot injection, he found that if the receptor occupancy by PET was between 70 and 80%, that was the sweet spot, and that when you gave higher doses, you would actually increase the number of extrapyramidal symptoms or side effects. When you went lower, it was not as efficacious. And the only exception to this is the mystery clozapine, where we still don't know exactly why it shuttled down. In a second, I'll show you where this curve has shifted over with newer drugs like aripiprazole. This is aripiprazole. It used to be called OPC then, but it's actually, this is Abilify. And this is a study that was published a few years ago with Katie Bezier and now Katie Smith from Otsuka at that time. The baseline raclopride scan, and this 2 milligrams per day in normal volunteers after 14 days. You can see there's a large blockade. And what we found is, of course, this curvilinear occupancy. And this is the expected dose range as predicted by Lars Fardy's earlier work. But what we determined was that the clinically used dose for Abilify, or aripiprazole, here it is, aripiprazole, was actually closer to 90 to 95%. So why is it that aripiprazole required this level of dose, whereas other antipsychotics, such as first and second generation antipsychotics, only required 60 to 80%? Well, the answer, I think, may have already been addressed in part by Professor Arvid Carlson, who has developed his own compound, and both aripiprazole and his compound are believed by many to be partial agonists of dopamine, as well as having dopamine D2 and other antagonism. And in the presence of such dopamine partial agonists, you have to push the dose higher. But the advantage of the partial agonism is that the number of side effects that occur at the higher doses seem to be a little lower. And so PET scanning can explain both the lower dose used by first and second generation drugs, the slightly higher dose that might be used in drugs like aripiprazole and other dose drugs such as in second and third generation, where there are more than dopamine that are factors such as serotonin and other things. And we won't have time to go into that today. One of the areas that I think is extremely important to our society because of the important role of marijuana and THC, there's a great debate about its role in medical marijuana, but we also know that there are still definitive issues that give us concern in terms of its widespread use. And so I'm not making a political statement. I'm simply indicating that we now have the tools to investigate the cannabinoid CB1 system. And this was done in collaboration with my colleague Andrew Horty, who helped invent these compounds. This is the THC and the CB1 cannabinoid system. And cannabinoids are tightly related to dopamine, and there are a number of studies showing the effects of cannabinoids causing dopamine changes, which of course ultimately relate to reward. We also know from the work of many, here's a study from D'Souza, showing that patients with schizophrenia have enhanced sensitivity to THC. And we also know, of course, that there's an epidemiologic study, including those by Robin Murray and others, that earlier use of THC may result in some increased epidemiologic evidence for earlier probability of psychosis in some populations. In any event, the point is that the THC has a two-edged sword. It could very well be very important for medical marijuana and for oncology studies, but it also has other issues. But it also can be useful therapeutically, as I'll show you, in areas like schizophrenia. So right now, there are multiple tracers used for studying CB1, and this is the one we developed, which is now called OMAR. And I wanted to use this time to just explain to you how PET tracers are developed briefly. The way it happens is you have a hypothesis. In this case, we can measure cannabinoids. We have to come up with a chemical development, like I showed you with Bob Danielson did with N-methylscriptone years ago. And then we can either do iodinated or technetium, which is called single photon emission tomography. I won't have time to talk about that today. Or we can use carbon-11 or F-18, which we develop in our in-house cyclotron. We then have a preclinical development that has to go through rodent biodistributions and an initial estimate of radiation dosimetry. These tracers have exquisitely low radiation burden, and in all the research studies, the amount of radiation is well below what radiologists in the United States, including people like me, are allowed to receive occupationally. We are at no additional risk. We have no additional morbidity or mortality than any other medical specialty. In fact, and so these are extremely low dose and essentially minimal radiation burden. But nevertheless, we have to determine this in animals. And then we go on to test these if they seem to work in animals in non-human primates. All of the primates that we study have been around for 10 or 15 years. They are very well taken care of, and we simply do a mild anesthetic to image them, but they're very predictive of what goes on to a human. And then if they look promising in, say, in a baboon or primate monkey study, we can then go on to develop the requirements from the FDA and the local Human Subjects Committee, including quantitation using kinetic mathematical modeling to develop these traces, and we do it either with PET or with SPECT. Here's an example with the cannabinoid OMAR, where you can see the regional distribution in mice, and this is in brain regions important to cannabinoids, and these are areas that do not have as much activity in cannabinoid binding, and then you can see the ratio, which is some indication of specific binding. And here, with self-blockade or with other drugs such as CB1 antagonists, such as rimonabant, we were able to demonstrate, both in the unblocked and blocked state, the specificity in multiple brain regions, as you can see here, cerebellum, cortex, hippocampus, etc., and thalamus. And in this case, rimonabant is a drug that was developed originally in Europe and was never received the go-ahead here, but it was very close to a drug used for schizophrenia, and we worked on derivatives of that with companies for schizophrenia. So it has been useful. These rimonabant derivatives have been useful as a potential antipsychotic, and they have also been useful for reducing eating urges, and has been a target for obesity, and it's still an important target. So the cannabinoid system has many medical beneficial aspects, which can be examined with PET. And this shows the next step. These are the PET data that we have here, correlating it with the known binding of the tracer in post-mortem human autoradiography. So you see they seem to correlate reasonably well. This is a non-human primate study of OMAR. It was called OMAR subsequently. That's the original JHU number. And these are PET and MRI studies showing the binding to putamen and other areas in the axial slice in the baboon and in the sagittal slice in the baboon brain. And then in the presence of rimonabant, 1 milligram per kilogram, you can see there's the significant blockade of the tracer in both these slices. And then once we were able to establish that this tracer would work in the non-human primate brain, we were confident to proceed with the requirements to go into humans. And here's an example of the intense uptake in the cingulate cortex in the human brain. We found that a measurement of the binding is called the volume of distribution, and there was a regional specificity expected throughout in normal volunteers. And then we also found, as is the case with most compounds, and we were fortunate to show in 1984, we demonstrated in vivo the first suggestion that D2 receptors might fall with age. Now it seems to be the case in almost all neurotransmitter systems between the ages of 18 and we go up to as much as 70. And we see this in multiple brain areas, in globus pallidus, cingulate, frontal cortex, and these are just the prediction limits associated with that regression line. One of the interesting findings we had, though, was when we studied patients with schizophrenia, we noticed that when we used the brief psychiatric, the BPRS, the brief psychiatric subscale for negative symptoms, that is anhedonia and lack of motivation and other issues, which is the most challenging area for developing schizophrenia drugs, that we were able to see that the PET binding correlated inversely with this, suggesting there was an important relationship between CB1 receptors and negative symptoms, giving us a clue for the next step of what might be helpful in some of the drugs for schizophrenia. Now, I want to quickly go through the metabolotropic system, because this is a very important area in neuropsychiatric disorders. And we know that the glutamine and glycine system have many connections, and glycine itself is important, and I'm going to talk about that intertwined with glutamate. Glycine has been linked through the NMDA receptor to schizophrenia, and many people try to increase glycine by giving glycine surrogates across the blood-brain barrier. But the problem is that they don't go across the blood-brain barrier well. The mGluR5 system is one of the most best developed systems, along with mGluR2 and 3, but mGluR5 is the one that we now can study with PET, and it's important in schizophrenia, depression, and other disorders. mGluR5 is actually a target, which many of you may know in Fragile X, including mouse models of Fragile X, one of the autistic spectrum syndromes. Reducing mGluR5 expression rescues Fragile X syndrome in mice. So measuring mGluR5 is very important in development. And here are a number of compounds that are available. We work with Ricky Waterhouse when she was at Merck, and with the Merck group on developing, through an NIH grant, one of these FPEP compounds, which was a derivative of what Merck had, and then we took it to the public space through an NIH grant, through NIMH. And we also looked at C11ABP, which is an mGluR5 compound developed in Zurich, and from the Novartis company and the University of Zurich. And here you see Fragile X. In our own Fragile X model at Hopkins, we showed that Fragile X was increased compared to the wild type using rodent PET studies. Most importantly, with Lyle Osterwell and Jeff Rothstein of the ALS Center at Hopkins, we looked at ALS mice, and you can see mGluR5 surprisingly has an elevation in ALS mice, which is especially bad in chronically ill mice. So we're doing this in humans now. And then this is the mGluR5 system in normal humans. Now, as I said, the glutamate and glycine system are linked, but most importantly, instead of trying to get glycine to cross the blood-brain barrier, glycine transporters are being used to block and increase glutamate. And so to measure this for so-called target engagement, we work with Adili Baroni at Roche and his colleagues in Basel to develop two PET ligands. This was the autoradiography that showed these compounds would work. We studied two of them at the same time in Papilionubis baboon brain, and we picked one. And the one that we picked went on to be studied preclinically. And it turned out that when we did an occupancy study, the occupancy that we thought matched what was done in animal behavior studies was about 40%. And that 40% occupancy seemed to match directly with what they found was the optimal delay to match test preclinically in Roche. We then went on to develop a human compound, which you can see here, for the glycine transporter. And then with the Roche colleagues, we did occupancy studies with bidopyridine, RG1678. And here are different studies shown in neuropsychopharmacology showing that at different dose levels, including shortly after the administration of the drug and then several hours later, we were able to demonstrate a dose response curve, as you can see here. Here's in one brain region, and here's the dose This is the dose of the drug orally, and this is the PET occupancy. And what's most important about this is, again, the 40% occupancy that we looked at seemed to match at that time the results they were getting in phase 2. That is, the phase 2 studies matched it. So in a sense, both PET and occupancy work. So finally, I'd like to briefly talk about amyloid, because this is very important, because this is the next frontier. And the first study that was developed was the Pittsburgh group, who developed radioactive dyes with the Pittsburgh compound B, done about a decade ago. And using this, Susan Resnick and others at the National Institute of Aging have done longitudinal studies showing at different ages, cross-sectionally, the increase of amyloid binding. And then longitudinally, many studies, and this is just one from the NIH-NIA, the Baltimore Longitudinal Study A, showing that even over a period of just a couple of years, there's an increase of amyloid accumulation that can be seen in people at risk for Alzheimer's and mild cognitive impairment. Now, F18, 110-minute compounds have been developed because they can be distributed to places that don't have cyclotrons. So here's 3 of them, and the one that we first developed at Hopkins with Avid, which is now part of Lilly, was AB45. This is the first one developed by called AB1. This is the second one. This was one developed about the same time by GE, flumenamil, first in Europe and then in the US. We studied it here. And this is the one we developed, and you can read about it in the Journal of Nuclear Medicine. This is a patient with Alzheimer's, and this is a normal control, showing the white matter uptake, but all the gray matter cortical uptake in patients with Alzheimer's. So these are now used routinely for the Alzheimer's Disease Neuroimaging Initiative, number 2, ADNI, for ongoing federally private-public relationships with pharma. And so this was picked in part by our collaborators at Avid because it was a nice point where the Alzheimer's and the controls, after about 40 minutes, showed a nice separation. And we also studied GE, not only the Avid compound was correlated with post-mortem studies in those patients that passed away shortly after scanning, but also we worked with flumenamil and GE correlating in vivo brain biopsies used for people with normal pressure hydrocephalus. And then the last 2 slides, just to mention that TSPO ligands have been used for neuroinflammation by my colleague Tom Ghilardi, who's now at Columbia, including brain injury and neuroinflammation. And finally, I just wanted to mention that we were fortunate to receive a planning grant from the Brain Initiative, the NIH, the White House, NIH, and this is a series of collaborators. We are now studying in vivo neurotransmitter modulation in real time. Remember I said it took somewhere between 60 and 100 minutes to scan a typical human or non-human primate. We're now working with the University of Copenhagen and University of Connecticut and others to develop voltage-sensitive dyes which release fluorescent light, which can give us measurements of, ideally, electrical activity in the millisecond range. So here's an example where light is shone in, and then it's redshifted, and we can measure that through external detectors. And working with Tony Grace, we hope that we can extend the known electrical activity to neuropharmacologic changes, which Dr. Grace is an expert on. So what we envision might happen is something like this. Now circle back to 1967, where they had these very fancy detectors for blood flow and xenon, and we might actually have something like this, but instead of using PET, we use optical tomography. So in summary, I just wanted to thank my many collaborators and all of you for your time, and I'm now ready to take questions.
Thank you, Dr. Wong, for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You submit them by typing them in the Q&A box, which can be found by clicking on the green Q&A button lower left of the presentation window. And we'll try to answer as many questions as we can. Our first question, Dr. Wong, what is the future of PET imaging in regards to temporal and spatial resolution? Do you think that we could use PET imaging in developmental studies safely?
I would like to ask the question about developmental studies. I presume they mean developmental patient groups. Is that the question about developmental studies?
The person who asked the question did not specify, Dr. Wong.
Abnormalities. What I did not show is we've had a number of studies involving Rett syndrome, Lesch-9 syndrome. We're collaborating with people on a number of other developmental disorders, including autism. At the present time, the local rules and to some extent the FDA is more reluctant to study people below 18 years, although some studies have been carried out with reduced radioactive doses at the NIH by Judith Rappaport and others. The last 3 slides that I showed you from the Brain Initiative does not involve any radioactivity. So it's possible that if we were able to, over the next 3 years, get this to work, these dyes would not involve any radioactivity and might be useful in developmental disorders.
Thank you. Our next question, how early can we diagnose the early stages of dementia and Alzheimer's disease using PET imaging? And can we differentiate frontotemporal dementia versus Alzheimer's?
So regarding the first question about early stages, there is a large literature that's growing throughout the world that there may be populations, not only people who have family histories and ApoE, certain ApoE genotypes, where we can detect these people very early on. Even with mild cognitive impairment, the amyloid imaging is allowing us to understand this better. It's not a perfect measurement yet, like most of these things, but there's a worldwide effort to try to do early detection. And in fact, the Baltimore Longitudinal Study of Aging Studies I just showed you has a number of studies that have illustrated these that early stages can be determined. And I'll direct you to many of these studies because if you just PubMed search it, you'll find literally thousands of recent articles on this field. It's not, I can't give a definitive answer yet because it's still a work in progress. The question about frontotemporal dementia versus AD, it's not a perfect relationship yet. We think that when we develop tau imaging agents in the future, this may help. Certainly, fluorodeoxyglucose has been used for frontotemporal dementia, but with amyloid, tau, and FDG, it should be much more, we should be much better at being able to differentiate FTD from Alzheimer's. So that's a very good question.
Thank you. Our next question. What is your impression of the glycine site partial agonist GLYX13 for depression? It's in phase 2 clinical trials right now, I believe.
I'm afraid that I can't answer that because I'm not familiar with that glycine site partial agonist at this time. I haven't worked with it, and I don't know anything about it academically. I think the main issue is that with glycine transporter type 1, there's not only the Roche compounds that we published, but there's other tracers that are available to look at the glycine transporter 1. And I guess the question I would ask Pamela Thompson is does it bind to the glycine transporter site 1? And if not, it's a little harder to say what we can do with it. Furthermore, my imaging studies that I illustrated are more focused to dose ranging as opposed to efficacy.
Thank you, Dr. Wong. Unfortunately, we are out of time. I would like to thank the audience for their interesting questions and participation in today's event, and our speaker, Dr. Dean Wong. Today's webcast will be available for on-demand viewing for the next few months. You'll receive an email from LabRoots alerting you when this webcast will be available for replay, and we invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Bye.